So good morning, everyone. My name is Szymon, and indeed I am a psychiatrist trying to learn from you about the modeling. And it is a big pleasure to be here and to present for you my first modeling approach. I'll be talking about the schizophrenia and the psychosis. So it's thematically linked to the previous talk. And as you know, the schizophrenia can clinically manifestate with different groups of symptoms. We include the positive symptoms and negative symptoms. And among positive symptoms, we've got the delusions that you heard a lot in the previous talk. And we also have hallucinations, mainly the auditory hallucinations. So beyond that, we know that schizophrenia is a disorder of imbalance excitation inhibition ratio in the brain through the cortex. We also know that schizophrenia is associated with many disturbances in neuroimaging, including fMRI signals. And probably because of that, it has been a successful target for many whole brain simulation studies based on this kind of data. I've listed some of them here. Probably part of the audience can spot their names here. So basically what was found is a little bit messy because depending on the exact population, some studies reported reduced excitation in the schizophrenia brains, while others basically the opposite, so the elevated excitation inhibition ratio, and probably it reflects the heterogeneity in the category of schizophrenia themselves, but also can be influenced on the exact pre-processing steps, etc. So mainly the studies that found the elevated excitation inhibition ratio was dealing with the global signal alteration that I will be talking today. So regarding the symptoms themselves, we know that this inhibition, mainly in a form of altered interneuron upregulation, is strongly implicated in the pathophysiology of positive symptoms, including hallucinations. And we've got many lines of evidence that support that, including the data from the genetics, from the molecular biology, and also the evidences from the computational psychiatry field. And here we've got the two very great studies. First of all is by Rick Adams, co-authored also by Professor Princeton. And it was a DCM study that dealt mostly with EEG data, but they also considered the small networks of resting state fMRI data. The second one is the whole brain modeling approach by Kinnett Wengler. And basically, both of these studies have shown that this inhibition is indeed implicated in the hallucinations. However, regarding the fMRI data, they were based mostly on the very small networks. So in this work, I try to expand on these results and test whether this association between the disinhibition and hallucination can also be reproduced by the study that is focusing on the large network statistics, like the whole brain's functional dynamic connectivity, etc. And the other objective of our work was to test whether this disinhibition relationship with hallucination also hold up when the excitation is limited to the physiological bounds. So we know that the schizophrenia is not related to the epileptic status, etc. So there should be a limit to the excitation that we can hold up in this disorder. So I used the OpenFMRI data from the UCLA. Oh, sorry. I take the data from 45 participants diagnosed with schizophrenia. I have also the matched healthy control group, and the hallucination severity was measured by the standard scale of SAPS. So yeah, we were modeling the OpenEye resting state fMRI data. I used the denoising strategy that preserved the global signal in the data because of this relation to elevated excitation inhibition ratio. And the main statistic of interest was the functional connectivity dynamics matrices calculated using the sliding window technique with the 60 seconds windows and 2 seconds step. So in terms of modeling itself, I used the model called the reduced Wang-Wang, also known as Deco-Wang dynamic mean field model, in the recent implementation by the Herzog and colleagues. And this implementation has a very convenient property that instead using iterative adjustment to the feedback inhibition control, it uses the linear approximation to it. So basically in this implementation, the local feedback inhibition values are for every node set as the multiplication between the global coupling, the structural connectivity strength of each mode, scaled by some factor alpha that allows the excitation inhibition ratio to be elevated or decreased, but still with working at least partially the feedback inhibition control mechanism. So the alpha and the global coupling was the three parameters in this study, as I said. And yeah, so the main modeling fitting target was the Kolmogorov-Smirnov distance between the distribution of the upper triangles of FCD matrices, average across 100 random initialization of the model. And we used, first of all, we used the classical neuroimaging fitting method, which basically meant for each participant to achieve the lowest scarce distance, so the best fit between the simulated and empirical data. And since I was really interested mostly in the correlation with some behavioral measures, I also used something called phenotypic fitting method, which was introduced, I think, like 2 years ago by the Young and colleagues. That basically, instead of optimizing for the best signal matches, we can optimize for the highest correlation between some model outputs and the behavioral feature that we are interested in. Yeah, so here we can see the results from single healthy participant. As we can see, basically the lowest KS value are achieved for the balanced excitation state, and the FCD matrices are reproduced quite well. And here are the main results of the study, mainly the results from the phenotypic fitting. And we can see on this figure that this is the landscape of correlation. And basically we can see one single gradient here, starting from the upper left to the right bottom. And basically this is the rising gradient of excitation in the dynamic mean field model. All the blue color here means that the correlation with the hallucination were positive, meaning that the model in this state better reflects the condition of people with low severity of hallucination. So we link the underexcitation to the low severity of hallucination. And quite opposite, the red one, it's a negative correlations, which state that the models using overexcited states better reflects brain dynamics of people with high severity of hallucinations. And the highest numerical value were achieved in the moderately overexcited state here. And those correlations were adjusted for the motion scanner, age, sex, etc. Here we can see the numerical results from the landscape. Basically, this is the highest positive correlation from the highest blue point. And here is the highest negative correlation, the more reddish point on this map. As we can imagine, they are separate quite nicely by the design, the blue and the red distributions. Regarding the empirical data, so here we've got the pooled populations from the schizophrenia group and the healthy control group are not separate really nice. And basically the difference between those two populations were not significant in any part. So we did some technical checkings. We used the same method, but with the shorter window time. The results were exactly the same. We also used unadjusted correlations. We also used the Pearson correlation instead of Spearman's ones. And basically all of these steps have reproduced the same results. So next, we want to know whether our results are specific to the hallucination themselves. And here we've got the phenotypic mapping with the positive symptoms, so the total SAPS score, and it looks basically the same. The effect sizes are little bit lower than for the hallucinations, but generally the gradient, etc., looks the same. So it is something that we could expect given that hallucinations are a main component of the positive symptoms themselves. However, from the negative symptoms, so the total SANS score, none of these correlations remain significant after false discovery rate correction. So they are really small, and the peak of negative correlation is slightly shifted for a less excitated state. So we can separate the negative and positive symptoms in this way. So beyond the phenotypic fitting method, I also use the classical neural fitting method, and basically the results told exactly the same story. so that the hallucination is related to the elevated excitation in the model. However, if I used only the classical fitting, I will miss the degeneration between the two parameters. So from the phenotypic fitting map, we can easily say that it doesn't matter how we get the overexcitated state. It doesn't matter if we modify the local inhibition on or the global coupling. However, when we will be using only this classical fitting, the significant correlation was only with local feedback inhibition and basically no correlation with global coupling. So that was the gain. Obviously, it would be much more interesting if the results goes beyond those single gradients to be more diverse, but probably because of this degeneration, it was only this one thing that we can spot it. So obviously we've got some limitations. We all know that this type of model fits a relatively low order properties of the signal. I can use it here because I know that the schizophrenia is known to be associated with altered global signal. However, we're still not sure if the global signal and their alteration is really neuronal or non-neuronal. Also, the sliding window method themselves have got the limitations that, like windowing the time series can, of course, bring back the regressed out signal, so increase the noise in the data. However, we tried to deal with that. First of all, the correlation between the mean motion and the hallucination wasn't high. And next, we've taken the nuisance signal and put through the same pipeline, and basically we didn't see the same results, so there's negative correlations. So I think that it's, at least part of it, it's covered. So obviously we also use the homogeneous model that didn't include the hierarchical status in the cortex, so probably I will try to do it maybe later. And that's all. So basically our results support the very large line of evidence that hallucinations and positive symptoms in schizophrenia are indeed associated with some kind of disinhibition in the cortex. Thank you very much.